Let's take a look at our Chapter 11 notes. This will be the first half. There's another audio notes that has the second half in them. So first, let's look at the Mendel's inheritance of human traits. So things that we talked about Gregor Mendel, we're going to now put into our human traits. We've talked about Punnett squares already, and we know that Punnett squares give us what the chance of having certain phenotypes and genotypes are. But when we have a pedigree, it's the actual description or tree of what happened and when they happened and to whom they happened to. So it really is a graphic representation of your genetic inheritance of a family or a couple. There are different symbols that are used to represent males and females. There you have different symbols that represent individuals who are affected or unaffected by a particular trait. A carrier is heterozygous for a recessive trait that does not have the disease in their phenotype, but does carry the allele so they can pass it down. On this slide, the things that you need to know for the test is you need to know what a pedigree is. You need to know that males and females are XY for male and XX for female. And you need to know what a carrier is, that it's somebody that does not show symptoms of the disorder, but can pass the disorder down to their children. Here are some of the representations that we look at when we look at pedigrees. The big ones that we're going to be looking at are males and females. Males are always square, females are round. If the individual is affected by the disease or is ill with the disease, they're usually colored in completely. If there's half color in, then they are usually heterozygous for recessive, which means they are a carrier, that they can pass it down. The Roman numerals represent the generations. If they are married, they are connected together by one. The children that come off of them come down off of the perpendicular line that goes along with them. So recessive disorders, and you need to know what this is, you need to know that they are disorders that appear or are expressed when the genotypes are homozygous recessive, which means we have two lowercase letters. They can't be heterozygous. To have the recessive trait show, both lower cases have to be there. Some examples of recessive disorders are cystic fibrosis, and this occurs when we have thick mucus in the lungs and the digestive tract, and it starts to accumulate. People who have this usually don't live a full life. Tay-Sachs disease, we had talked about this before on our last test. Lipids aren't broken down in the brain cells, and the lipids or these fats accumulate, and the brain function then thus declines and eventually leads to death. It's very common in those of Eastern European or Jewish descent. The children who have this do not live very long, and they usually die a pretty painful death. Another example of a recessive disorder is PKU, phenylketonuria, and this is when the amino acid phenylalanine cannot be broken down. What happens is it accumulates and mental retardation can form. This can be treated by a diet low in phenylalanine. So this is something that they try to catch very, very, very early when the babies are first born. So those are recessive disorders. We also have dominant disorders. And these are, you need to know what these are for your test also. These are disorders that are expressed when the genotype is heterozygous or homozygous for dominant, which basically means you just have to have one capital letter to get these. So you have to realize that these are probably a little more prevalent than some of the other diseases we talked about. You will also notice when the set of first examples here, cleft chin, widow's peak, hitchhiker's thumb, mid-digit hair, all those things that we worked on with the lab that we did, you'll see with those that most of those aren't life-threatening because they can be passed down so much easier. Though there is Huntington's disease, which is a result of the breakdown of certain areas of the brain. There's no treatment for this, with it being a dominant passage down. You only have to be heterozygous dominant to have obtained this disease. When we think of Mendelian genetics, we think of those simple rules that you get one gene from mom, one gene from dad, and you do get one gene from mom and one from dad. But most of our inheritance does not follow this simple pattern established by that. We can have things that are called incomplete dominance. This is when we have heterozygotes that have an appearance that is in between the phenotypes of the two homozygous. When you have incomplete dominance, you always use capital letters, and you get the phenotype that appears is going to be a blend of the two phenotypes that you start off with. You need to know on this slide that heterozygotes have an appearance that is in between the phenotypes of the two homozygous. It's like being blended. An example of this is when I have red flowers that are crossed with white flowers. We would use big R for our red flowers, big W for our white flowers. And then when we put those into a Punnett square, we would get a blending of pink flowers between the two, and that's incomplete dominance. And to work out that problem, I would have a red flower, big R, and up on the top, big W on the side. And if I would cross them in each one of my four squares, I would get big R, big W, big R, big W, big R, big W, and big R, big W. And we know that big R is red, big W is white, and RW is pink. When you're doing these, it's really important that you establish what each characteristic stands for, what a letter it is, and what is the blended letter. Remember, the blended combination has to be a combination of letters you used. You can't change them in the middle. You can't say L is for long tail, N is for no tail, and then to make short tail, you can't make it an S. You can make it a capital L, capital N. So keep that in mind. It's a common mistake that kids make. Another example of when it doesn't follow the Mendel rules, has different rules, is when we have multiple alleles. So every trait that we have discussed so far, it only takes 2 alleles to show the phenotype of it. But, and you need to know this for your test, some traits have more than 2 possible alleles. Even though we can have more than 2 possible alleles, you still only get 2 of them that's going to show you the trait. But what then ends up happening is you have a much broader spectrum of what the phenotype can be. An example of a multiple allele is your blood type. We have A, B, and O blood groups. The letters that are used for this refer to the carbohydrates and proteins that are found on the surface of the red blood cells. If they are present, it tells you if the blood is type A, type B, or type O, or type AB. So the possible alleles for the blood type is, and we always use I for blood type, is I with an A at the top. That would be that A carbohydrate is present. I with capital B at the top. That means the carbohydrate B is present. The lowercase I means there's no carbohydrate present, and that would be O blood. Both A and B are dominant over I. So if I is present and one of the capital A's or B's are there, it's going to be the one that shows up. So here's your possible genotypes and phenotypes for blood type. So I can have, as you see, my type A can be I, capital A, I, capital A. Type B, I, capital B, I, capital B. Type AB can be I, capital A with I, capital B. But I can also have blood type AB, capital IA with a lowercase I, because A is dominant, or B can be capital IB, lowercase I, can be type B blood. The only way I can get type O blood is if I have 2 lowercase i's together. Okay? So an example of the multiple allele problem is you can look on your worksheet that we did, and I want you to look at one of your blood type 1s and work yourself through that. Another example of when they don't follow the Mendel rules is codominance. So far we've done incomplete dominance, we've done multiple alleles, and now we're going to look at codominance. If you notice when both the capital IA and the capital IB are paired together, both alleles are expressed in the blood type AB. So they're both there. This is codominance. Both alleles are expressed in the heterozygotes, but both alleles are dominant, right? So you need to know that for the test. What that basically means is both alleles are going to show up. An example of codominance is what we call sickle cell disease, an example of this. And in sickle cell, you get a breakdown of the red blood cells. The red blood cells aren't round anymore. They become more sickle-shaped. And what happens then is they start to clump together and they clog up your vessels. Accumulation of these sickle cells can accumulate in the spleen. And then they can pass through your heart. They can be painful. You can get fever, brain damage, weakness, kidney failures as these goes through and block up these different vessels that they can block up. And this is kind of what they look like. The normal ones, you have these nice round cells that can move smoothly through. These other ones, the second one, the sickle cell anemia, you have some of these cells and they start to clump up a little bit, but when they're severe, where you get these turns, etc., in the vessels, they really clog up in those areas. So when we look at our genotypes and phenotypes here, again, in this scenarios, in codominance, we always use capital letters again. So we're going to use N for normal and S for sickle cells. Capital S means you have sickle cell disease. NN means you have no symptoms. NN is no symptoms. You're healthy, you're normal. SN means you're healthy, but you have some of the symptoms at high altitudes. And then you also have it, you can pass it on to the next person. Sickle cell is interesting in that about 1 in 10 African Americans is to carry a sickle cell, but the disease is very rare in Americans of other ancestries. And this is because the gene is so common. If the effects are so, you want to know why they're so common if the effects are so serious. And what is the reason for that is what we call heterozygous advantage. That means the heterozygous for a particular trait have some advantage over the homozygous don't. So if I'm a carrier of sickle cell, which means I'd be heterozygous, right? I'd be capital S N. In this case, that heterozygous gives you a resistance to malaria. Why is that interesting? Because malaria is very common and prevalent in Africa. So that is why it becomes a heterozygous advantage. If you are heterozygous for it, you may not have the full-blown disease, but it does help you with a resistance to malaria. You're less likely to get that, which is something that you could acquire in those locations and die for it. In those locations where malaria is prevalent, heterozygous those people with heterozygous for sickle cell, capital S, capital N, tend to live longer and produce more offspring, which is what we think about. So here we go in the African regions. This is where malaria is very common. And so this is why people of that descent are more likely to have it. So if I did a cross between these, I have a sickle cell carrier, capital S, capital N, put that on the top for mom, and they marry somebody that has sickle cell. So then I would do my cross. I would have capital S in the top left. And the next one I would have capital S, capital N. In the bottom left, I would have capital S, the capital S, capital N. 50% of my offspring will be carriers and 50% will have sickle cell. Another example of possibilities that do not follow. Oh, first let's talk about sex determination before we talk about sex-linked traits. There are two possible sex chromosomes in humans. They are X and they are Y. Females are double X, males are XY. That was on our last test, and a lot of you had a real problem with that. Remember that it's the sperm that determine the sex of the offspring, because moms, or the females, can only give an X away. Males can give either an X or a Y away. So if I did an example of this, I would have XY at the top for mom and X for dad. XY on the top for dad, XX on the sides for mom, and then on here I would have XX, XY, then XX, XY, which means I would always have a 50% chance of having a girl, 50% chance of having a boy. So what are sex-linked traits? These are genes that are located on the sex chromosomes. And most sex-linked genes are located on the X chromosome. the phenotype can be seen in both males and females. So it can still show up in both males and females, but the genes are carried on the X chromosome. You need to know and understand that the phenotype can be seen in both males and female. A few are located on the Y chromosomes, but phenotype can only be seen in males if on Y can only be seen in males. So make sure you know that. If it's on Y, it can only be seen in males. Which means most of them are X-linked, but there can be some Y-linked, but they can only be seen in males if they are Y-linked. So one example of sex-linked disorders is color blindness. So you need to know that that's an example of this. And this is when the light, you have malfunction of the light, it's sensitive eyes in malfunction of the light, the sensitive cells in the eyes. Your red-green see gray tones instead of red and green. This involves several X-linked genes. Normal vision sees over 150 colors. Color blindness or color deficiency see fewer than 25. So a colorblind father cannot pass it on to a son. A colorblind father cannot pass it on to a son. You need to know that. You need to understand that. And that's because the colorblind father is on the X gene. And the only thing that a father can pass on to a son is a Y. That's how the son becomes a son. So a colorblind father cannot pass it on to the son. So can females be colorblind? This is an example right here of a colorblind test that we'll do in class. Can females be colorblind? The answer to this is yes. Possible alleles are big X, X with a big B, which is normal, X with a little b, which means you're color deficient, which means it's recessive. So here's your possible genotypes. Big X, big B, big X, big B, normal female. Big X, B, big Y, normal male. Big X, big B, big X, little b, female carrier. Big X, little b, big X, little b, color deficient female. Big X, little b, and big Y is color deficient male. So make sure you kind of know all of those. And then you can do an example of this. So take a normal female and cross it with a color deficient male. Write it down and see what you get. Can males be carriers of X-linked traits? No. Why not? They can't because they when they give it away, there is usually a trait on the X chromosome of the mother that can cover it up. So it's not going to be the one that is going to give it. And it usually shows up in males and they give Ys. Okay, they give Ys. So an example is hemophilia, X-linked gene. You get excessive bleeding. When injured, lack clotting may bleed to death. After minor injury, excessive pools of blood. You need to know that it is an X-linked gene. Okay. Another example is your Duchenne muscular dystrophy, progressive weakening and loss of muscle tissue. Usually death occurs around the age of 20. Next thing that we want to talk about is polygenic inheritance. And this is things like height, skin tone. Those are two examples of those, height and skin tone. So polygenic inheritance, this is the additive effects of two or more genes that appear in a single phenotypic characteristic. And so you need to know what that definition is. These characteristics tend to occur on a continuum, which means like a bell curve. All right, so what are some examples of this? Height, for example. You usually have very few short people and very few tall people, and everybody else kind of falls in the center of that bell curve. It's like drawing a straight line near the bottom, come up and make a bell, and then come back down. We're going to do an example of that with some Post-it notes out in the hallway during this chapter so that you can see them. All right? So those are things such as the skin tone height. And this is what a bell curve looks like. You have few people at each end of your spectrum. There are also other things that can affect the phenotype, and these are external influences on your phenotype. These are things that can also occur with your genes. This is our roll dice activity. So when we do that, and I want you to think of this when it comes in. So number one, we have external influences on our phenotype. These are environmental things that can affect the expression of certain genes. Some examples of these are temperature, light, nutrition, and different chemicals. Right? So those are things that the phenotype can be controlled by. Phenotype can be controlled by things like temperature, light, nutrition, and chemicals. All right? It can also be controlled by internal influences on phenotypes. And the internal influences on phenotypes are gene expressions that can be affected by things like hormones being present, structural differences, age, those kind of things. Those are the kind of things that can affect the genes, um, which then affects the phenotype, right? So know that these external, internal influences, what they do is they affect the gene itself, the genes, and then those genes are then going to affect what the phenotype itself are. These can cause that. So, complex inheritance of human traits and karyotyping. This is where the second half of the other note starts. So we'll end right here.